Today, I'm going to talk about mainly population dynamics related to rats. And part of this relates to some global trends of what we can discern are patterns happening in cities around the world. And part of this, we're going to zoom into a individual city or individual cities and individual city blocks. Okay, so we have heard about rats. You guys generally have the background. I'm kind of in a room of experts, so I don't think I need to go into much detail, except to say that there are 70 different species within the genus Rattus, and we are really only focusing on one or two for a meeting like this. Rattus norvegicus, which is the only one that we would have consistently, at least in a place like New York City, and also many of you guys in other cities, especially south of here, are thinking about roof rats as well, Rattus rattus. You guys know these are globally distributed. Kaylee mentioned that earlier. They started dispersing when we had trade and movement of trade products around the world. And they are the classic urban exploiter. They do really well around humans' nests when they make their own nests. And part of that is that they're adapted to multiply. And you'll see a lot of themes, especially in the middle part of this talk, related to their ability to reproduce really, really well. So I guess we should take it at face value that at a summit like this, we are all sharing information with the goal of limiting their numbers. Now, several people already outlined that limiting them to zero is not a realistic goal, but it's one that we need to appreciate. We're trying to get the population size down in these conversations, and that's sort of how I have geared the information that I'm going to talk about. And as Kaylee and Chelsea mentioned earlier, we've had a long war against rats and we've been unsuccessful for the most part. I'm going to show some numbers about long-term trends here in a little bit, but this is a article from the French press from 2016. And the mayor of New York mentioned this earlier, Mayor Adams. So rat attack, Paris declares war on cities' exploding rodent populations. There, the lessons were learned more quickly. 7 years later, what did they say? Paris needs to learn to live with rats, mayor concedes, okay? So something that's taken decades, maybe in New York, or even centuries to come to terms with, the mayor of Paris took 7 years. Okay, so why we care? I don't think I need to go into detail because other speakers in the morning session outlined this very nicely, but usually I'm talking to audiences about disease risk. A lot of what we do in my lab is related to disease detection, but I think Kaylee and Maureen and Chelsea did a great job handling that and discussing those details. Another big part of this is economic damages, whether it has to do with damage to infrastructure, but also related to food consumption and food contamination, either in an urban setting or an agricultural setting. And then lastly, Kaylee mentioned really nicely, went into detail about the mental health implications. I think that's an important theme that really hasn't been talked about at all until the last 4 or 5 years since the Vancouver Rat Project started publishing some data related to this. So I'm going to divide this talk into 3 sections. And we're going to start with a global perspective, what's going on in cities around the world. Then we'll zoom into a city block scale picture of rodent ecology, rat ecology, and population dynamics. And then lastly, we will talk about if we can make predictions city by city using some sort of modeling. I have no idea how to use this long space, so I apologize in advance. I'll see what I can do. I can barely see the screen up here. All right, so let's talk about these global trends to start out with. First of all, we need to not just take the assumption at face value that rat numbers are increasing, right? That's something that's talked about a lot, but we don't have any long-term scientific tracking of this. And as a scientist, that drives me nuts because we talk about this all the time, but we don't have any solid data to back that up over more than one city at a time. So we're going to talk about what evidence we do have and what data is actually available to answer this question. A little hint, it's imperfect data, but it's what we can use to answer these questions or provide some insight. Because when scientists don't provide this insight and don't have these numbers available, the media, I know there's some here, nothing personal to you guys, they're happy to fill in this vacuum and talk about all sorts of increases in rats, when in fact some of these data may be flawed at best. So we as biologists have real reasons to think that rat numbers should be increasing. It's a reasonable expectation. Some of those reasons have to do with increasing human population densities in cities and what that means for food and resources available for the rats. I can't see this side at all, so sorry, I'm going to keep looking this way. Cities are growing, so that means there's more urbanization, which means there's more urban habitat, which is presumably something that they are at this point well adapted towards. And also climate warming. So this is a species that is presumably limited by cold weather. Both species, in fact, are limited by cold weather, at least their above ground activity and their ability to forage. right? They're small mammals, so maybe climate warming is feeding into their ability to be active for longer, reproduce more often, have more pups across the year, potentially increasing these populations. Okay, so what did we do in this study? Well, as we mentioned, there are trade-offs in these sources of data, whether they're collected by professional inspectional services looking for active rat signs versus using public complaint data like 311. But we have what we have. And so this is what we used for this study. We went to, first of all, we looked at the 200 largest cities in the United States and got the data that was available for at least 10 years in systems where the data intake collection system had not changed dramatically over that 10-year period. So that's at least 10 years. We wanted to expand this beyond the United States, so we worked with collaborators in Tokyo, Amsterdam, and Toronto to get data in other cities as well. I think this is an indictment about the way we collect data, but in the end, the number of cities that met our standards for collection was only 16 cities, right? We looked at more than 200 cities, and only 16 had suitable data to do an analysis like that. That's not good. That's a message I want this sort of community and this set of stakeholders to take note of and say, we need to do better in terms of collecting systematic data. Okay, and there are a big set of authors on this paper, and it's very important. First of all, there's 6 or 7 or 8 in this room. Thank you very much, collaborators. But it's a nice interaction between academics and municipal employees and also researchers that are not necessarily part of academia as well. But all those voices and those perspectives came into this study. So I appreciate everybody that contributed to this study in very important ways. And I mentioned that public complaint data is imperfect. I think most of the people in this room will acknowledge that. But it is correlated. So Maureen, who spoke earlier this morning, did one of the first studies to try to correlate actual trapping data with public complaint data. And so that data is from Chicago in a paper she published in 2018. And then just earlier this year, a group in the Netherlands published this data from 3 different cities across the Netherlands and also found that there was a good correlation between trapping data, active rat signs, and public complaints. So we can take some solace in the fact that there is an association between public complaint data and actual rats. Importantly for this study, we're tracking numbers of rodent complaints over time within a city. And so any biases, which we know exist in 311 data, should be baked into that and consistent over that time period. So it shouldn't bias anything within a city by city perspective. Okay, so I'm going to cut to the chase and show you guys the data. And what we're going to look at first is something called a Mann-Kendall trend analysis. So that's basically just a time series regression that accounts for some of the autocorrelation from one time point to another. And what I have here is an empty plot, not interesting yet, but what I want us to note is that this Z statistic, this Z value on the x-axis is our important metric. So that red line is sort of the magic point. A Z value of 0 means there's no trend. The rat numbers aren't increasing and aren't decreasing. Anything to the right of that red line means that there's an increase in rat numbers over the study time period. Anything to the left is a decrease. What do you guys think the data will show? All right, I know what the media thinks this will show, and yeah, they're mostly right. So 11 out of the 16 cities did indeed have significantly increasing trends of rat numbers. Only 3 cities had significantly decreasing trends in rat numbers. And then there's Dallas and St. Louis there that didn't have significant values one way or the other. That's the distinction there. Okay, so I don't know, about 70% of cities do indeed have a statistically distinguishable increasing number of rats as measured by 311 proxy data. So this is over the course of at least 10 years. Some cities had as much as 17 years of data, so between 10 and 17 years of data. Okay, so that's nice. That's a trend. That's interesting. I'm not a statistician, so I'm a biologist. I want to know, are there some biological things and environmental things that we can pick up as being responsible for those trends? And so we thought, these are our hypotheses, mostly ranked in the order we thought they would be playing a role. We thought human population density would be important. We thought the mean minimum temperature in a city might be important because, again, colder periods of the year should see a lot less rat activity for both species, less foraging, less opportunities to have the body condition that would be suitable for breeding. Next up, we thought long-term temperature trends might be important, right? If a city is warming because of climate change and climate warming, we might expect the active period for these rats to grow. Maybe they can come out a little bit earlier every year. Maybe they can start going down and be forced to be in their burrows a little bit later in the year. So giving them a few weeks extra on either side of this. We thought economics are important because early analyses we had done in other studies indicated that socioeconomics was important in terms of rat abundance and what was going on there. I think the previous speakers this morning did a nice job talking about that. And then urbanization. We know the land cover that rats have access to and the infrastructure they have access to are important in terms of their day-to-day daily life and ecology. All right, so I'm going to start off by showing you a relative importance analysis. It's just a fancy term for a bunch of regressions that are grouped together and they can account for variation within those. And the y-axis here is the percent of overall variation in these trends that is explained. And on the x-axis, it's just going to be 5 categories, those environmental variables that we just walked through. And what we can see is that it was the change in temperature over long-term baselines that explain most of the variation. I'll dive into that here in a little bit. But the other ones that were significant were urbanization, the amount of urbanization or the inverse of vegetated land cover within a city, and then human population density. Surprisingly, GDP as a measure of overall socioeconomic health of a city or economic resources and mean minimum temperature made no difference at all, wasn't correlated at all. So it didn't fit our hypotheses there. All right, so let's dive into one of those variables, change in temperature over time period. So first of all, each one of these points is data from one city. So there's 16 points up there. On the x-axis, we have temperature change over a 50-year baseline. And on the y-axis, we have that Mann-Kendall Z statistic. So what that's saying is as we move towards the right on the x-axis, those are cities that are warming faster, warming more. Those cities have faster increases in their rat numbers. Again, using 311 as a proxy. Okay, the size of the symbol has something to do with population density there. Okay, so there is a climate change link to increases in rat numbers, at least from the data that we have. That urbanization part of it. So here on the x-axis, now we have the percent green land cover. So as we move left now on this plot, on that x-axis, we have cities that are more and more urbanized, less and less green space, less vegetation cover. Those cities have faster increases in rat numbers. All right. So let's come to this question. Are rat numbers increasing? Yes, generally they are, but it's not universal. There are cities where that's not happening. There's a climate change connection, and I can see absolutely no silver lining to this. Caroline, you and I were talking about this earlier. This is essentially sort of an additional pressure on the system that cities are going to have to deal with if climate warming is allowing rats to be more and more successful over time. It's something we need to acknowledge, and we need to build into things like plans and budgets and all those sorts of things. Is this an argument for more green space? Well, I like green space, so I want to go out on a limb and say that, but we need more fine scale, fine resolution information on what habitats the rats are using, but that's a correlative results from this. And then this certainly suggests that there's no long-term success in our war on rats, which Chelsea did a nice job talking about earlier today. Another thing I want this group to think about, because this is the group of players that would be making a difference and are talking to all these different cities and all these different agencies within different cities, but what can we learn from these 3 cities down here? I'll be honest with you, I don't know much about Louisville, but I know we have someone from New Orleans here. We have 2 people from New Orleans here. And so we can talk to Claudia and Timmy about what they do in their cities that seems to be successful. I call them a gold standard city because of the way they do things. And one of the things they do very well is education. They have a lot of education outreach for communities in New Orleans. Tokyo, I work with Yasu. He's the one that was able to get the data there, Kiyokawa. And one thing he talks about is cultural expectations of cleanliness. A bag of garbage on the street in Tokyo would never happen, okay? Would never happen. And so that's a cultural aspect. They have very high expectations for their food service establishments. They have very high expectations for the average citizen to take care of their garbage and not allow it to be on the street and serve as a food resource there, okay? And I shouldn't have forgotten number 1 there, sanitation, right? That's what we're trying to implement here in New York City with the containerization laws. Okay, so let's zoom in a little bit. Let's focus on individual cities and in particular, block by block analysis of what these rats are doing and how that changes over time and what lessons we can learn from control that becomes part of that, or control efforts that are implemented as we track these populations over time. Okay, so here we've decided to go all in on rat control, but that's not enough. To be able to be successful, we really need to have a solid understanding of basic rat ecology and rat demography. As an ecologist, it hurts my heart when we talk about implementing programs and plans without having a thorough knowledge of basic rat ecology. So one question I have is, do we have that? So what I want you guys to do, if you haven't already scanned that QR code, let's do this. I'm going to try to jump out of PowerPoint for a second. Lord help me. And let's hope that this works in the online format. So I'm going to ask you guys 4 different questions. If I can you guys see questions already? All right, let's go to this first one. Don't be swayed by peers when you see their answers appear up here. Make sure your answers are independent. So let me. Too quick. Okay. Sorry, guys. Sorry. Was that Neve? I could tell the voice. Is it appearing as a big screen for you guys? Oh no, that thing came up. All right. Oh wait, here we go. Okay. Yes, okay. Okay, someone asked what species. So what time of year are rats most numerous? So we're talking about a temperate city like New York. So we would assume it's mostly Rattus norvegicus, the Norway rat. Let's see what the experts here. We have a room full of experts. I'm trying, yeah. Oh, there we go. Yeah. It's getting stuck on the second question. Can some people still see the first question? All right, let me see if I can, sorry guys. Thank you. I feel like a stand-up comedian up here, where the hecklers start coming out. All right, if you're stuck on question number 2, I'm sorry. We'll just think deeply. Telepath your answers up here to question number 1. All right, so it looks like, so the question, in a city like New York City with a temperate climate, what time of year do you expect rats to be most numerous? And we have mostly in the summer, June through August. We have some people saying spring into early summer, and then the second most saying sort of early fall into the middle of fall. Okay, all right. Let's hope this works, guys. Let's go to the next question. For Norway rats in our region, so temperate region, what time of year has the highest reproduction rates? Is it the same time as you expect the population to be highest? And I'm only going to provide answers for at least 2 or 3 of these, not all 4. We'll have to think about this over conversations later today and tomorrow. No, yeah, even if you can't see all this responses on this screen, hopefully you can see them on your phones. You can see all options. Okay. All right, so for the question, what time of year has the highest reproduction rates? It looks like we have the vast majority, well, not the plurality, saying March through May, and then next up is in the summer, June through August. It's okay. Yeah, that's this is based on what's actually happening in the population of rats. So if we were perfect trappers, and if 311 data was perfectly mirroring what's actually going on in the population, that's what I'm imagining here. Okay, so it looks like we have spring and through the summer. All right, next up. Larger female Norway rats produce more pups than smaller females. We think about body condition, age, dominant structures, as much of that exists in female Norway rats. After I get done with this talk, I'm going to record these for perpetuity. I'm going to include them in future talks, all your answers. This is what the group of experts said. Let's see how close they were. Okay, so about 3/4 say that that is false. About 1/4 say it's true. Last but not least, whoop. All right, I cleared it, so you might have to answer again. Which age class of rats are most likely to enter traps and take the rodenticide bait that's been one of our primary strategies over the years? And yes, weanling is a real term. So someone who is just weaned from the mother. Not someone, some animal. I see we have very little faith in the male gender, okay? It's fair, it's fair. Okay, so it looks like about 2/3 of folks believe the juvenile males are dumber and less experienced, or I should say more bold and experimental, shall we say? All right, so thank you guys for humoring me in this poll. I probably will use this for future reference, but we all have a sense of where we are and the variation of our understanding. So let's get into some detail that might shed actual light on this. It worked. We're there. Okay, that's perfect. Yeah. Okay. All right, so that's our understanding as a group of experts in the year 2024. But if we want to dig into this literature, unfortunately, there hasn't been much appetite to explore hardcore population ecology of rats for about 60 or 70 years. So we're going to dig into a little bit of old school literature on ecology and demography of these rats in what I call the golden age of urban rat research. In front of what other audience could I use that term, the golden age of urban rat research, than this one? So we're going to go back to Baltimore, Maryland in this golden age. So many of you guys may be aware that there's a lot of historic data, a lot of historic studies coming from Baltimore, and it all emanates from Dave Davis's lab. So he was at the Johns Hopkins, what was then called the School of Hygiene and Public Health, for about 15 years in the 1940s and 50s before moving on to Penn State and then ultimately NC State. But what he did is what many of us aren't willing to do today, where he would go out and measure numbers of rats, active rat signs, things like that, in many different blocks across Baltimore and a few other cities as well. And he would track those both just as standard conditions within a city, but also you see that top image is pre-sanitation intervention, and that bottom image on the right is post-intervention. And so we'll talk a little bit about some of these control strategies that he worked around. But this is what one of the data collection cards look like. This data is from 1945. So John Emlen, that's the JE there, he went out and he studied this block in Baltimore for about 2 hours. And he would do this regularly. And he would go and he would measure things like, are there fresh fecal pellets? Are there active burrows? Are there trails clipped in the vegetation? All those things that we look for here in this audience when we go out and look for evidence of rats. Fresh tracks, infestation level. And so if you do that repeatedly and track it over time, you can get a nice time series of what these populations are actually doing, whether you're trying to intervene with poisons or some sanitation practices or not. You can see how these populations are changing. And you'll notice here that he came up with an estimate of rats in this block of 170. He didn't say, oh, I think there's about 1 rat per human on this block, which is often a number we cite. But for his era, he was using state-of-the-art population ecological mathematical models to estimate population size. And he did that for many different blocks across his different studies. And by prolific, first of all, he was in his 30s when he started this position, and I think he transitioned positions in his early 40s. He puts me to shame. I feel very embarrassed. He would publish like 10 papers a year on this stuff. So very, very prolific, very good science. So this is what some of his data would look like if we try to track the cycling of these rats across an entire year. Now, obviously, looking at data plots today is a little easier to do than back then. So I'm going to try to summarize this data. This is for 5 or 6 city blocks on the left and another 2 blocks on the right. But generally, what he saw across these systems is a bimodal distribution across the year. So if we summarize all of his results across all of his papers, We have time on the x-axis and rat numbers. Now, this is just a sketch that I did. This is not scientific data, but I'm trying to summarize the patterns that he consistently found across his studies. And so what he found is consistent with what some of you guys were saying in the poll survey. Rat numbers generally tend to be lowest in the winter, rise in the spring into the early summer, dip a little bit in the summer, and then pop back up in the fall, and then come back to some wintertime, I wouldn't say equilibrium, but low point there. I want to juxtapose this with what our 311 data look like. So this is data from a study that we did that Michael Parsons led, and this is New York City 311 data over the course of about 8 years leading up to COVID lockdowns when things got a little weird. But does that look bimodal? No, that's one peak every single year, and that peak is during the summertime. So some of those biases related to 311 data, that shows up between the population ecology and the 311 public complaint data. So just something to take into account. All right, so those cycles don't just come out of nowhere, they come out of the breeding ecology of these species. Okay, so here is data from Davis 1953. This is one block around a warehouse in Baltimore. And we're looking at time again on the x-axis and now percent of females that he trapped that were pregnant. All right, again, not the easiest data to look at. So I tried to make it a little easy by highlighting the curve there. Does that look bimodal? Do we see two peaks? Yeah, it looks like two peaks there. There's another curve there with the X marks there. That's actually the percent of females that are lactating. So I don't have time to get into that, but we can take into account the number of females that are pregnant versus how long they're lactating. Are they supporting pups? Are they having multiple breeding episodes per year? That sheds some light there. But I want to highlight this bimodal number or percentage of females that are pregnant, the two separate peaks there. So there's our spring peak and our fall peak that fits that bimodal distribution of population size. And he looked in other cities, or he drew data from other cities and found similar patterns in places like Norfolk, Virginia, San Francisco, California, and then England just outside of London, but still an urbanized type habitat. And we see those bimodal distributions. Now, maybe they're shifted a little bit because the climates are different in those different cities, but generally that bimodal distribution is still there. Okay, so I've tried to have some signpost points here that maybe we can draw some practical insights from this. So practical insight number one is, are lethal control interventions more effective during these peak periods of either rat population size or percentage of females that are pregnant in these populations than at other times of the year, right? Because I don't hear a lot of conversations about distributing our control efforts differently at different points of the year. I kind of hear about these programs and they happen the same. We can do it when we can get staff out on the field. That's not a criticism, but maybe it's not fine-tuning our strategies based on the ecology of this organism. Okay, so let's talk about control and how that feeds into that. Bobby, you and I were talking about some of this data earlier. So this is a residential city block in Baltimore, a different one. Okay, you'll see lots of different Baltimore residential blocks that were surveyed by Davis and his team. And so on the y-axis, we have the number of rats, and on the x-axis, we have time again. So we're looking here at a period of about 6 or 7 years. Okay, and there's a few different time points that we can take note of here. This green zone here is intense control that was entirely rodenticide based, and it actually did work. It led to local eradication. Okay, population went from about 100 to 0, and it stayed about 0 for just shy of 2 years, even after they stopped control. But then about 2 years later, you had a handful of intrepid rats. Maybe we call them like the Baltimore pizza rats, okay? Really went out there and tried to do their thing, came back in, and then you have a period of about 6 months or 12 months where you have exponential population growth. Now, if you think about bacteria on a culture plate, on a petri dish, we know about exponential population growth. Well, this is a small mammal that does the same thing. If you give them the resources, you have males and females in that population. And then it exploded. And after that, there was this period of, I don't want to call it stability, but the population is going up and down, bouncing around what we call a carrying capacity in ecology. It seems like that particular block in Baltimore could support, roughly 100 or 125 rats. Sometimes there were a few more rats, sometimes there were fewer rats. But these are classic population ecology growth curves that we can draw inference from. And this is where the term boomerang effect or rebound effect came in, because consistently the population would bounce up and down, especially around these control intervention campaigns. All right, so even more hard to look at graphs. So I'm going to break this down for you guys. This is two other blocks in Baltimore. One block didn't really have, it was sort of treated haphazardly, so I'm not going to think much about that data, but I'm going to highlight this block right here. So this blue curve refers to one of the blocks. And what I want you guys to notice, each one of those P's there, they're going to highlight here with an orange circle. That's when rodenticide poison bait was put down. And if we get rid of that other curve, just to make it simple to look at, does it seem like the rodenticide poison bait worked? Short term, it seems to have worked consistently, 5 times at least. But what happens a few months after that bait pulse is removed? It jumps back up until that 5th time where the city decided to implement some sanitation strategies, which in this case had to do with implementing fines for residents that had debris and other harborage type garbage around their lot. They had to containerize their trash, and they also had to cement up earthen basement spaces. And so that's followed by a period of at least 2.5 years of consistent retained population decline in that case. Okay, and then they ended the study, so we don't know what happened after that, but that's a good sign for sanitation, right? That's a good piece of evidence. And we see something like that in modern studies too. Now, there's not a whole lot of them, but one of the cities I work in is Salvador, Brazil, with Federico Costa and his team, but they did something similar in 2014 and 15. So again, time is on the x-axis here, and on the y-axis is active rat signs, proportion of samples that had active rat signs. Okay, and so in this case, when they implemented, again, rodenticide-based campaign, they had a decrease in the population size, or at least active rat signs, for 12 months. And then afterwards, the rats bounced back. So there was that rebound. So we're seeing that in modern times too. All right, so practical insight number 2, rodenticide use alone is never going to be enough. I think everybody in the room appreciates that part. And habitat remediation is a necessary part of IPM. Okay, so how does body size fit into this? That's another important part of a small mammal. How big is it? How many pups can it reproduce? How often can it reproduce? So just to show you some data here, this is Davis and Hall, 1951. On the x-axis, I wish these axes were inverted, but they're not. So percent pregnant females and body weight. So I highlighted the important one here in blue. The larger the female, the more likely they are to be pregnant, okay? And if we look over here on this plot, I've simplified a little bit, it's hard to look at, but they looked at 4 different size classes of female rats. And what we see is here, the largest rats are there in red at the top, and the smallest rats are in blue, and the intermediate ones in between. The y-axis here is percent pregnancy, and again, across time. Now, I realize these little lines I drew are something my 8-year-old could have done with crowns, but hopefully that makes it easier to look at. But what I want to highlight here, there are 2 peaks in pregnancy, right? We saw that in previous data. That's not surprising. But what is surprising is that even the small females, which shouldn't be able to reproduce as much or produce as many offspring, they even have their moment. And in the fall, they're reproducing at the same rates as the larger females. So size does play into this. Larger females do breed more often, are pregnant more often, do have more pups, but even the females can do this. All right, so what I wanted to show you guys is some data on body size distribution. So this is from at least 30 different collaborators around the world we're collating the data from. And these are size distributions with size of the individuals, males and females, on the x-axis in grams, and these different cities that we have data sets for. This is just Norway rats at this point. You see New York City here. There are 2 data sets, one from Matt Combs and his trapping during his PhD, the other one from Kaylee Firth in her trapping when she was at Columbia University. So I wanted to first of all say, congratulations, New York City. You do have the largest rats in the world, at least from one of those data sets. But more than being a point of civic pride, it also means that there's more of an uphill battle, right? Bigger females, we've seen from that previous data, will breed more often, be pregnant more often, and produce more pups. So that's an added challenge that maybe you have in a city that has these larger rats. Okay, so those critical insights are from 70 years ago for the most part. But remember that this is a species that is experiencing natural selection, some of it of our own making by applying rodenticides and exposing them to natural selection related to who can survive or who can avoid eating that bait. So these rats have probably evolved in that time. So does our lethal control drive evolution in important ways? So I'm going to talk just briefly about a side project related to the genetics of this. And this relates to that campaign of control that I mentioned from Salvador, Brazil in 2014 and 15. We took advantage of that and went and collected rats before they started that campaign, immediately after they finished that campaign, and about 7 months later, 1 or 2 generations later. And we wanted to know, how do these campaigns impact the genetics of these urban rats? Okay, so as I mentioned, the 3 sampling time points, in the end, we collected about 240 rats and took their genetic samples and sequenced those. And then we analyzed those for 3 different valleys. In Salvador, the focus of the study is like a favela community that really struggles with leptospirosis infection rates. And so here we looked at 3 different valleys and at 3 different time points. And what we expect, based on just basic evolutionary biology, is that there should be what we call a bottleneck effect. If you try to knock a population down over and over and over again, it should take a lot of genetic variation and make it a little genetic variation. It should send that genetic variation through a bottleneck. Okay, so we would expect more inbreeding and a shift in population genetics. Okay, and when we look at, for example, on the y-axis here, genetic population size of those 3 valleys before the eradication campaign, that's the blue bars. And then in all 3 cases, after we hammered them with rodenticides and knocked their populations back, their genetic population size, or their genetic variation, dipped by 60 to 75%. That's a huge loss in genetic variation. When we think about inbreeding, on the y-axis now we have relatedness, which is a measure of inbreeding. We can see in 2 of the 3 valleys, inbreeding went up significantly. We all know it's not good to inbreed. That must have some impacts on the rats. And then last but not least is population genetic signature. Don't worry about this x-axis. It doesn't make intuitive sense. Just know that these curves, again, blue is the curve, the genetic signature for the pre-eradication population, and red and gold are after. We can see in all 3 valleys, there's a dramatic shift in population genetic signature of these populations. Okay, so what does that mean? What are our implications for the people in this room? First of all, suppression campaigns do often have effects, but they tend to be short-term. But clearly, there are substantial genetic impacts on these populations. And we, as a research community especially, have to follow up on that and look at what the long-term impacts are. It could go either way. Evolutionary biology would say that having a genetic bottleneck should have bad deleterious effects for that population, because you don't have as much genetic fodder to deal with changes in your environment. New rodenticides coming through, perhaps. But the other side of this is that we have been nailing these things with natural selection, and the only ones that make it on the other side are ones that are really well adapted to deal with that chemical, that active ingredient that you've exposed them to, or maybe behaviorally in avoiding our bait box and our trap. So practical insight number 4, long-term genetic viability of these pest populations, and this doesn't just have to be for rats, it should absolutely be something that we start to integrate into these integrated pest management plans that we're designing and thinking and talking about. And as much as I was harping on the media earlier, I wrote this article and the editor for this had a different idea in mind for the title. So she won. Super rats or sickly rodents. Our war against urban rats could be leading to swift evolutionary changes. And we ignore this at our own peril. And it's not just anticoagulant resistance, but it's also other things, including behavioral traits. Okay, last section. So we're going to zoom in on individual cities and think about just briefly how we can make predictions about where best spatially we can target our control for these animals. Okay, we already talked about how we can predict where these guys are, or what pieces of data we can use. We talked about public complaint data and how it's imperfect, but is correlated in important ways. Data from municipal surveys, so what Caroline's team is doing in terms of sending trained professionals out there to figure out where active rat signs are present. Maybe we have a little more faith in those, a lot more faith. And then there's those of us, Maureen, thank you for using the term nerdy rat scientists. That's oftentimes what I refer to us as. So we're in a safe space here. So, the academicians in this world kind of have a role to contribute data to this as well. And in terms of prediction, there are new techniques like machine learning, and I think Boston's trying some machine learning. John's team up there, Derek, your team. But I'm going to talk about tried and true habitat suitability modeling for just like 5 or 6 slides. And so there's a lot of history in ecology with habitat suitability models. So let me walk through what that is real quick. It takes months, but all you need to generate a habitat suitability model is occurrence data. So where are the organisms on the landscape? For us, that's professionals saying, yes, we saw the thing here, we didn't see it here, or 311 reports. Then you need environmental variables, so habitat variables, and we do this all in GIS. And then we use statistical models to look at the degree of overlap between any given environmental variable and whether the animal is there. And those statistical associations give us these suitability output models. Okay, so if I cut to the chase, this is what it looks like for Manhattan. And Caroline, thank you for clarifying this earlier when we were talking. So this is data, and I should say this is in collaboration with Matt Combs, who's down here in the front row as well. So in 2015, the Department of Health undertook this pretty extensive effort to look for active rat signs in some areas of focus, chronic infestation zones, but also the larger island of Manhattan. And so this is what the habitat suitability model looks like from that 2015 Department of Health data. Okay, we'll dig into this a little bit, but I mean, the gist of it is that the warmer colors indicate more suitable habitat. So we went back and said, okay, it's great to have professionals out there, but that doesn't happen very often. What if we use the 311 public complaint data to create the same sort of model for the same time period? And so I'll come back to that table here in a little bit. This is what it looks like for 2015 311 reports. And it's remarkably consistent. I find that really, really encouraging that the public complaint data gives us essentially the same predictive picture of where the rats would be likely to be as when the professionals went out. And if we look at this table over here, the nice thing about habitat suitability models is that it gives you a % contribution of each of those environmental variables that you put in the model and how likely they are to explain where the rats are. And what we see with the professional survey is that, probably no surprise, the density of food service establishments is really important, as is human population density. When we look at the public complaint data, it's pretty similar. Like the ranked order is roughly the same. Social vulnerability index, which is heavily weighted towards socioeconomics, is a big part of that as well. And there are some zones that stick out. People in New York City, especially, you know, professionals here working on this day in and day out, you'll recognize some of those what are now rat mitigation zones or the chronic infestation zones. They show up pretty clearly in this model. So it seems to have good predictive power. So I'll be interested to hear what Caroline's going to mention earlier. She's done some more sophisticated modeling of this with her team as well. So we'll learn more about this. When we zoom out to the entirety of the 5 boroughs of New York City, here you'll see the time period here is the 1 year just before the sanitation pilot program starts, because we didn't want that data to be biased. It's a little hard to see any specific neighborhood here on this map, but what I want to point out is that human population density, for reasons that I don't understand, jumps to the top of the most important variables in explaining where the rats are in these citywide models. So that's something that I'd love to talk with the Department of Health about, see if we can make sense of that. I imagine it has to do with places like Staten Island or Eastern Queens that really are much, much lower density residential than the rest of New York. And I don't want to go one more slide without acknowledging that basically Dave Davis did something similar 70 years ago. So I'm using fancy scientific tools and GIS mapping and blah, blah, blah. He kind of did the same thing where he was looking at zones of infestation back then. And you'll notice that 70 years ago, some of those same areas were chronically infested with rats. So this is a decadal problem that we still haven't figured out. Maybe it's a wicked problem that we can't solve, or that, you know, the uphill battle. We're like Sisyphus pushing that rock up the hill. All right, and we're doing this for more than just New York City. So we're working on data from DC. I see Gerard Brown over there. So data from DC, Salvador, Brazil, and we're expanding this to Europe as well. So that's Vienna, Austria up there, and we're trying to get data for Barcelona as well. Because our ultimate goal is not to just do it city by city, but to generate a generalized model that even cities that don't have us nerdy rat biologists mucking around can use this data to make predictions about their own city. And this is not modeling, but I want to throw it in there as the last slide. So there's a very important pilot program going on in West Harlem. And I think, Caroline, you're going to talk about this in a little bit. But you could create really fine scale, sophisticated models of habitat suitability, even on a block by block basis, if you have somebody willing to go around and, you know, tabulate the number of boroughs and things like that. So far it looks like sanitation is moving things in the right direction, but we'll hear more about that from Caroline. All right, so all in there. Thank you guys very much. And I'm happy to take questions at the end if we have time later today.